what if the methylation of this particular gene happens, right? This segment of DNA undergo methylation. There is no mutation going on. There is no change in the sequence of DNA. There is no any kind of chromosomal aberration. Just because of methylation, this gene will become silent. Okay. Now, if I show you the structure of DNA, it is a very important structure. Okay, let me show you. If we observe the ultramicroscopic structure of DNA, here we can see this is a cell. The cell contains the nucleus. Inside the nucleus, this is condensed structure chromatin network as chromosome. If we observe the detailed structure of chromosome, it is the chromatin network that is wound around these sea green color balls. These are basically histone proteins. And if we observe this chromatin network more in detail, so it is a double-stranded structure of DNA. Means this DNA is wound around histone protein. And the tightening of DNA around histone protein play a very important role in gene expression. So the second way in which the gene can increase or decrease its expression is histones are a special type of protein around which DNA has been wrapped up. Loosening or tightening them increase or decrease the gene expression. If the DNA become loose, the gene expression decrease. If the DNA get tightened, so the gene expression increase. So this is something we need to understand that this time DNA is not modifying. There is no mutation going on. There is no chromosomal aberration going on, but still the gene expression have been affected. So this is the case of epigenetics. In another case, there are some chemical tags that the segment of DNA undergo chemical tag that cause the switch on or switch off over a period of time. Environmental trigger for epigenetic changes, it may be related to trauma, stress, nutrition, maternal care, certain toxic chemical substances, physical activity, aging. These are all the different types of environmental factors that can upregulate or downregulate certain type of genes. Children raised in a stressful environment and abusive environment show epigenetic changes linked to anxiety, depression, and stress activity. It has been identified that the fact they tend to experience more anxiety, depression, and stress compared to a child raised in calm environment. Epigenetics and inheritance are like after lecture 14, the concept of evolution, we will be discussing psychoactive disease, some different psychological disorders, where we will learn that how different type of psychological disorders, what are the traits and features of different psychological disorders, and what is the ratio of their heritability. Epigenetics and inheritance. Some epigenetic changes can be passed from parents to offspring. Like if parents have the capacity to trigger certain genes on onset of uh, certain indicator, so the same phenomena is happening in their children, affecting the trait across the generation. Famine exposed affecting grandchildren uh, birth weight. So if the children are born to the area where people are affected with famine, where enough food is not available, so even the next to next generation child birth weight will also be affected. There are some real life application under gene environment application. If we are talking about the heritability of intelligence in enriched environment, better teaching, economic stability, emotional support, they all are linked up together. In the same way, mental illnesses, personality differences, and addiction increase the vulnerability of an individual. And there are more likeliness of psychological disorder if the person is raised in vulnerable environment with deprived nutrients and basic hygiene is not maintained. So if we sum up all together what we have compiled so far, the behavior, how the individual appear and behave does not merely depend upon uh, the set of gene that individual has inherited, but gene environment interaction play a very important role. And another important thing that gene expression can be upregulated or downregulated due to certain environmental factors. So, Alhamdulillah, lecture 13 is completed. If there is any question, you can ask. Do you want to ask anything related to this lecture?
जैसे एग्जाम में ये किस तरह से आ सकता है?
Okay. First of all, uh, we might give you different case studies based on this topic. For example, we can give you the case study of two individuals, and suppose if the two individuals are mentioned with similar gender, and we say that it is a twin study, right? And we ask you to predict, is it a case of identical twin or fraternal twin? So obviously, if you do not have any much more detail about the individual, just on the basis of similar gender individual, you will identify that this could be a case of identical twins, right? And now you can defend that the behavior of the individual and the predictability, like if these twins are parted in different environmental condition and they behave differently from each other. So as both of them have same set of genes, they are 100% alike in their genetic makeup. It is the environment which is responsible for difference in their behavior. Okay. First type of question could be like this, or maybe we can give you the question that what are the ways in which um, the name different psychological studies that could be done to identify the gene environment interaction. So here you can suggest that uh, the different studies that could be done to identify gene environment interaction are, it can be twin studies, it can be adaptation studies, it can be the case of inheritance, or it can be related to epigenetics, right? Is that clear, Vika?
Thank you so much.
We might ask you between a different type of heritability. We can ask you different about of twin studies. It can be monozygotic twin, can be dizygotic twin studies, or it can be reared apart twins, or it can be the longitudinal twin studies, like we are studying twins over a period of time. So on basis of the situation that examiner is putting in front of you, you have to write the answer according to that situation. Define this, define that. Questions are less likely to be asked. For example, epigenetics, yes, it is a keyword. This could be asked by examiner, right? Certain keywords can be asked by examiner, but this topic is more logical and discussion based. Am I clear, Peter?
Yes, miss.
Okay. Now the next concept that we are going to discuss is genetics, inheritance, and theories of evolution. Please don't mark anything on the screen. Okay. When we were doing the concept of genetics case studies, so in that class, we discussed about the sex determination. And I told you about the karyotype analysis. If we take a cell of an organism, like it is a diagram that show karyotype is basically a diagram that show the arrangement of chromosome. As we can see, total 23 pair of chromosome. 23 pair of chromosome means total 46 chromosome. So this karyotype is basically a human karyotype. Okay, how can we identify whether it is a male individual karyotype or a female individual karyotype? On the last pair, I would observe that the last pair has one long chromosome and one short chromosome. This longer chromosome is X chromosome, and this short chromosome is called as Y chromosome. So karyotype is basically a pictorial illustration that show all the chromosomes arranged in sequence. It gives us more detailed information about an individual. We can identify the gender of individual. Even some chromosomal mutation can also be identified with it, with the help of this diagram. Let's suppose if I show you a karyotype analysis and I show you one extra chromosome at this place. So do you think it is a normal set of chromosome for a human, or there is some type of mutation here? Is it a normal karyotype of a human male, or this diagram is showing any type of mutation?
Normal.
Normal. Uh, since uh, there was like before I draw this red color chromosome, right? It was a normal karyotype analysis. But what if I have drawn one extra chromosome on 22nd position? This is not a normal DNA sequence. This is representing a mutation. What is mutation? The mutation is actually error in copying DNA. If there are two type of mutation. One is chromosomal mutation, the other is gene mutation. So if the whole chromosome is added or depleted, this is actually representing chromosomal mutation, right? So this karyotype, when I have drawn one extra chromosome, now this is no more a normal karyotype. This is not representing a normal human male. It is representing a Down syndrome male. Is that clear? On the basis of karyotype analysis, how can we identify whether it is a male individual or whether it is a female individual? If the last pair of chromosomes are exactly identical, then we will say that it is a male individual. Sorry, if both the chromosomes are exactly alike, then we will say this is a female individual. But if one is X chromosome and one is Y chromosome, one is longer chromosome and the other is a shorter chromosome, then I would say that this is a male individual. If we talk about the sex determination in humans, so okay, this is something important concept. Please listen it very carefully. If we are talking about the humans, so in humans we can see total 23 pair. 1, 2, 3, 4, 5. There are 23 pair of chromosomes. This is 22nd pair. This is 23rd pair. 1 till 22 pairs. These are called autosomes. The first 22 pairs are known as autosomes, and they are responsible for all the body characters. Means they are responsible for hair color, hair texture, your ability to learn, your ability to speak, etc. But if we talk about the last pair of chromosome, the last pair of chromosome is actually responsible for the last pair of chromosome is actually responsible for gender determination. This is called the sex chromosome. It is responsible for gender identification of an individual. Okay. So now, if the examiner is asking you the case that how we are going to identify what is the predictability that individual is, we are going to do the case study in which we identify that how gender of an individual can be identified. So, male at the time of sexual reproduction, male individual tend to produce sperms. At one time of sexual intercourse, a male tend to produce about 300 million sperms, and out of those sperms, half of the sperm contain X chromosome and half of the sperm contain Y chromosome. And in case of female, whatever the number of eggs a female make entire in her life, so all the eggs will have X chromosome, right? So at the time of sexual intercourse, one sperm is going to fuse with the egg. So now female is going to produce all the eggs that contain X chromosome, and male is going to produce half of the sperm contain X chromosome and half of the sperm that contain Y chromosome. Now, if I show you this checkerboard, so on basis of it, what is the probability of a female child? Every time fertilization take place, what is the possibility that the child born is going to be a male child?
Half.
Out. Guess what?
50%.
50% chance. 50% chance that a female child is born showing XX, and 50% chance the male child is born, right? So first of all, we need to understand that the gender identification, every time fertilization takes place, there is equal likeliness of a male or a female child. The second thing we need to understand, this is a very common social issue, that if in a society, if like in our society, if a male child is not born, so usually females are marked responsible for not giving birth of a male child. But scientifically, who is actually responsible for the provision of Y chromosome? Is it the female responsible for um, provision of Y chromosome for a male child? Who is responsible?
The male.
Maternal side or paternal side? From which side the Y chromosome is available?
Paternal.
Paternal.
It is the paternal side. So scientifically, a female cannot be blamed for not giving birth of a male child. This is a scientific and logical reason for which we need to identify that gender. I the process of fertilization is random. We have studied the concept of mitosis and meiosis, and we have learned that at the time of fertilization, it is at random that which gamete is going to fuse with it. It is neither the male individual fault nor the female individual fault. It is entirely a random process. Is that clear? Is that clear, Vita?
Yes, ma'am.
Okay. Now, the next important thing we need to understand, what is mutation? It is change in gene or chromosomal structure. Either a small segment of DNA. What is a gene? It's a small segment of DNA. When we say that mutation, what is mutation? Either a small gene is changed or the whole chromosome structure is changed. When I showed you this karyotype and I asked you that is it a normal male child or a normal female individual, so you identify that this is a normal individual. But what if at any position, at position 7, at any other position, I make one extra chromosome? Or in any case, if I delete one chromosome, right? This is also a possibility that one chromosome is deleted, extra chromosome is added, or one chromosome is deleted. In all those cases, we will call it as mutation. Now, mutation that occur in normal body cells, somatic cell, are not passed from one generation to another. For example, if a person had a tumor or abnormal cell division in any limb or in any somatic organ of the body, so they are not heritable, they are not passed on from one generation to another. But what if mutation occur in the gametes? If the mutation has changed the number of chromosome or structure of DNA within a gamete, so this is a case of heritable mutation. Spontaneous mutation can arise during replication or repair of DNA. These mutation, once we studied the concept of cell division, we discussed DNA replication happens under synthesis phase, and right after synthesis phase, G2 phase happens that makes sure that if there is any kind of error in DNA, it has to be corrected. But what if that error is not removed and passed on from one generation to from the cell, and the cell enter into the phase of mitosis or meiosis? Then the mutation will be passed on. Mutation can also be caused by exposure of mutagen. Scientific study indicate that there are certain environmental factors that increase the chance of mutation. For example, ultraviolet radiation, X-rays, radioactive particles, or gamma rays. There are certain type of chemical substances such as benzene, bromide, nitrous oxide. These chemicals are used in scientific labs. Exposure to these chemicals may lead to mutation. Gene mutation actually increase the amount of genetic variation in the gene pool. But remember, not all mutations are lethal. Some mutations can be favorable, right? When we think of mutation, it appears to be dangerous, but not all mutations are dangerous. Sometimes they just bring the variation. For example, the different eye color, the dimples on on the cheeks. This is a kind of this is a result of genetic mutation, but they are not lethal. Okay, there are type of mutation: point mutation, frame shift mutation, insertion or deletion, or chromosomal mutation. What is mutation? What are different type of mutagen? What are the different types of mutation? You like examiner might ask you based on mutation. It is something important. Okay, what can you understand from the word deletion? Delete means you are removing something, right? That section of DNA has been removed. Can you see this is a segment of DNA? And once deletion happens, can you see this particular mark segment has been removed? We cannot see this red marked structure. So if a type of mutation in which a segment of the DNA has been lost, this is called deletion mutation. Okay. What is duplication mutation? Duplicate refer to what? Copied amount, right? If you observe the diagram of duplication, can you see a certain marked segment of DNA has been repeated? Can you see in duplication a segment of in case of duplication, a certain segment of DNA is a certain segment of a certain segment of a certain segment of DNA has replicated itself. Now, if you observe insertion, so if you observe carefully, a certain a certain marked segment of DNA, it is darker. It is shown darker from the top and lighter from the edge. And if you observe carefully in inversion, it has been turned upside down. The segment of DNA has been changed, has been inverted. So all these cases are the case of point mutation where only one gene is being affected. Either it is deleted or it has been